And we are rolling. And I wanted to finish up talking about Haemosporidia, this last group of apicomplexans. There are quite a lot of species. There are some, sorry, I just heard the ding dong. There are some that infect birds, some that infect amphibians and reptiles. They are, okay, here we go. Yeah, we were talking of apicomplexans. There are some that infect birds. There are some that infect reptiles. I think there may be some that infect mammals and fish, but I don't know much about them. Far and away, the one that humanity tends to worry about most is the causative agents of human malaria in the genus Plasmodium. Just to recap, you get these by getting bitten by an infected mosquito, and that injects the infective stage called the sporozoites. In human malaria, these immediately head for your liver, and they penetrate liver cells and divide asexually repeatedly and turn the liver cell into a mass of cells called a schizont, and that's one of them there from a liver biopsy. The schizonts then burst to release the little swarmers, the merozoites. This part of the life cycle is called the extra-erythrocytic life cycle. Remember that erythrocyte means a red blood cell, extra means outside of. So this is the part of the life cycle that takes place outside of the red blood cells. And then the merozoites penetrate red blood cells. We're now in the erythrocytic cycle. And the merozoites penetrate, transform into trophozoites. The trophozoites, again, you have this stage with the big central vacuole and the nucleus pushed to one side. So this is the ring stage. And once mature, the trophozoites begin dividing. They create a mass of swarmers inside a red blood cell. The whole thing is called a schizont. That's one on the right. And the schizonts burst, release new merozoites, and this cycle can go on indefinitely, giving you the classic symptoms of chills, followed by fever, followed by a sweat. Once every 3 days if you have Plasmodium falciparum, once every 4 days if you have any of the other 4 species. In each round, a few merozoites transform into gametocytes. This is them in Plasmodium falciparum, looking a little like a banana, I guess. Other species, they might be more oval or round. That's the stage that infects a mosquito that takes a blood meal. So if a mosquito bites an infected person, the gametocytes enter the mosquito's body, and it's within the mosquito that the sexual phase of the life cycle takes place. Mosquitoes are the definitive hosts of Plasmodium, and we'll go through all of these stages and talk you through what happens. No sooner has the mosquito slurped up the blood, within about an hour, the gametocytes come in 2 types, micro and macro. These are analogous to eggs and sperm. And the microgametocyte divides into 8 slender little microgametes. On the left, you can see it caught in the act through a scanning electron microscope. On the right, you can see it happening in a blood sample. You can sometimes see this happening in a human blood sample if you let it sit for about an hour before you stain it. The process is triggered by the blood becoming a bit more acidic, and this happens naturally in blood that's drawn and then allowed to stand. So exflagellation happens, out come the microgametes, and they find the macrogametocytes and fertilize them, so we get a zygote. So they're the microgametocytes exflagellate. The gametes fertilize the macrogametocyte. You get a zygote, and the zygote transforms into a mobile stage called an ookinete. Remember, kinene, again, is Greek for to move. So the ookinete is the mobile egg, the mobile fertilized egg in this case. This is the only stage with a complete apical complex, and this burrows through the gut wall and will sit there on the outer surface of the gut. And this happens within a day. So here someone has dissected out a mosquito gut and stained it with the light microscope. And on the gut wall, you can see a whole bunch of ookinetes that have rounded up and formed, by this stage, they're called oocysts on the wall of the midgut, sitting there on the outside of the gut wall. That's what they look like under the light microscope. And that's what they look like under the scanning electron microscope. Now, the twisty things that you're seeing are trachea. Those are those air passages through which the mosquito can breathe, and they transport oxygen to all parts of the body. So that's the twisty filament-looking things. You can see them clearly at the lower right, but the things that are kind of bulging up and sitting on the outside of the gut wall, those are the oocysts. And within each oocyst, the ookinete divides into many sporozoites. This ought to start sounding familiar now. We're coming to where we can link the ends of the life cycle. When the oocysts rupture, they release sporozoites into the mosquito's gut. So there you've got a mosquito gut under dark field light microscopy. At the lower right quarter, you can see a number of these oocysts. They're the round things looking distinctive in that grainy mass that is the mosquito's gut itself. And they've ruptured and they've released all of these small little crescent-shaped or banana-shaped things. Those are the sporozoites. That's the infective stage. And that stage migrates to the mosquito's salivary glands. And when the mosquito bites a host, it injects saliva. The saliva's got an anticoagulant in it to keep the blood from clotting as the mosquito drinks it. Incidentally, the anticoagulant triggers an allergic reaction, which is why mosquito bites itch. But when the mosquito injects saliva, it injects sporozoites into a new host, and round goes the circle of life, and it moves us all. That was an awful lot to work out, and a lot of the history of parasitology has come from people working out life cycles. And as we'll see several times over, it's great if you can find a medication that will kill off a particular parasite. It can be tricky, though, trickier than it is for bacteria, because with antibacterial drugs, you can find a function that the bacterial cell has that your cells don't. Penicillin, for example, and its relative drugs like cloxacillin and amoxicillin and all that, they act by blocking the bacteria's ability to form peptidoglycan cell walls. Since your cells don't have cell walls, peptidoglycan or otherwise, your cells are not particularly affected when you get dosed with penicillin. That's what makes penicillin a remarkably safe antibiotic. The closer the pathogen is to you, the tougher it is to find drugs that will kill the pathogen but not have severe side effects on you. And so while I'm not saying we shouldn't try to develop anti-parasitic drugs, some of the amazing success stories in fighting parasitic disease have come from understanding the life cycle and finding a spot where you can break it, finding a spot where the parasite's vulnerable, where it's straightforward and fairly easy to block transmission. The guy that made this possible was a fellow by the name of Sir Ronald Ross. I might occasionally ask you test questions on personal names, by the way, but don't worry about memorizing dates or anything like that. Ross won the Nobel Prize in 1902. I think that might have been the second Nobel Prize awarded, because he worked out the Plasmodium malaria life cycle under some very difficult conditions, serving in India, which at the time was part of the British Empire, with the Indian Medical Service from 1881 to 1899. His original hypothesis was that malaria arose from intestinal poisoning, but he got a leave of absence to go back to England in 1894, and he studied with a guy named Patrick Manson, who had come up with a hypothesis that malaria was mosquito transmitted. And as soon as Ross got back to India, he started testing that hypothesis. And that's where he worked. It was very difficult work. Working out the stages in humans was fairly straightforward. He had no shortage of patients with malaria, and you could find the parasite and all of the stages of its life cycle in human blood samples pretty easily. That was pretty straightforward. Figuring out what it was doing in the mosquitoes was much more difficult. He had to dissect mosquitoes and extract their guts. You've already found that that's not always an easy process, even with something the size of a cockroach. Dissecting mosquitoes is much more difficult. He's working in India in the summer, which, if anything, is more hot and humid than Arkansas at the same time, and found that if there was any air movement in the room, his mosquito would blow away. So he had to turn off the ceiling fans, which were the only thing that the hospital had that made life even remotely comfortable. So imagine dissecting something the size of a mosquito in a room with no air conditioning and no ceiling fans in the height of summer. That's what he had to put up with. And Ross, by the way, was also something of a poet. And after seeing his patients in India, he wrote, "In this, O nature, yield, I pray, to me. I pace and think, and take the fevered hands, and note down all I see, that some dim distant light may haply break." Haply means luckily. The painful faces ask, "Can we not cure?" We answer, "No, not yet. We seek the laws. O God, reveal through all this thing obscure the unseen small but million murdering cause." He finally caught a break in August of 1897, August 21st. He paid a malaria parasite to let mosquitoes bite him and kept the mosquitoes, dissected their guts 4 days later, and was finally able to observe oocyst formation. These are his actual notes. Hard to read his handwriting, but you can see 21st August up there at the top left. And those are his drawings of mosquito oocysts, the same things that you saw on an earlier slide. And being a poet, he wrote this in his notebook: "This day relenting God hath placed within my hand a wondrous thing, and God be praised. At his command, seeking his secret deeds with tears and toiling breath, I find thy cunning seeds, O million murdering death. I know this little thing a myriad men will save. O death, where is thy sting? Thy victory, O grave." I don't know. I confess to a certain admiration for scientists that try to turn the wonders of the discoveries that they make into poetry. You know, it's kind of cool. Ross was not very cool about sharing credit because an Italian parasitologist named Giovanni Battista Grassi, who's shown here, had been working on malaria as well, not having to go to India because it used to be endemic in places like Italy. In fact, heck, malaria is an Italian word that we borrowed into English. Malaria is bad air, because people had noticed ever since the Romans that if you lived close to a stinky, fetid swamp, you were likely to come down with malaria if you lived in swampy areas. Malaria, of course, isn't caused by the air of a swamp. It's caused by the mosquitoes of a swamp. But they had noticed that correlation. Like I said, the Romans knew about this sort of thing. They knew that it wasn't healthy to live close to a swamp. Anyway, Grassi had worked out the life cycle at the same time that Ross was doing it. He worked it out independently and arguably did a somewhat better job of it than Ross did. The Nobel Prize Committee intended to have Ross share the prize with Grassi, but Ross may have been a poet and a scientist, but he was also kind of a tool. He accused Grassi of fraud and pulled some strings and used political connections and denied Grassi the Nobel Prize. Grassi would go on to make a lot more important contributions to parasitology, working with parasitic worms of various sorts. And we may get to see later some of the things that he did. But he probably deserved a share of the Nobel in 1902, and Ross cut him out because Ross may have been a poet, but he was also a jerk. It happens. Anyway, the reason knowing that life cycle matters is that it tells you what's a vulnerable point to break it. And far and away, the easiest way to break it is to keep yourself from getting bitten by a mosquito. Now, if you have been outside in Arkansas in the summer for more than 5 minutes, you know that's easier said than done. It was a major problem in World War II when U.S. soldiers were fighting in the tropics. I guess it might have been a problem in Italy, but it was particularly a problem in the war in the Pacific. About 60,000 U.S. soldiers died of malaria in World War II. The disease used to kill about 700,000 people annually, although that number's gone down recently, as we'll see. Mostly it's children under 5 that die. Adults can live with it, but if you're flat on your back 1 day out of every 3 from chills, fever, headache, and sweats, that tends to cut down on what you can accomplish. You can't work, you can't earn money, you can't take care of your house or your garden or anything like that. So it's an economic drain, even in populations, even for people that it doesn't actually kill. And there's an estimated 250 million cases of malaria each year. Now, the Army, of course, realized that, the way to stop infection would be to block the mosquitoes. So here you've got a World War II poster warning soldiers against going to bed with malaria-carrying mosquitoes. So there, I don't know, I just think this is kind of a cool poster, reminding you to sleep under a net, keep it repaired, and tuck it in. And since the 1600s, people have used a drug called quinine. Quinine was actually isolated from a South American tree called the cinchona. When the Spanish began exploring South America, they found that native people were using the bark of this tree against the South American form of malaria, which is a different species from P. falciparum. And specifically, it was priests in the Jesuit order that found this out and actually held a monopoly on the trade for a while. It used to be called Jesuit bark, the bark of the cinchona tree. The active ingredient in the cinchona tree is quinine, and quinine blocks the trophozoite's ability to break down hemoglobin inside red blood cells. Now, quinine has been mostly replaced by a more effective form called chloroquine. You may have heard of this because there's a derivative of chloroquine called hydroxychloroquine, which was proposed recently as a wonder cure for the coronavirus, which unfortunately it seems not to be. This family of drugs works by messing with the environment inside cells, which I think is why it was thought that it might stop the virus, because viruses are, of course, very different from malaria parasites, but viruses and malaria both have to reproduce inside human cells. And if you can mess with the environment inside a cell in just the right way, you might be able to block replication. Incidentally, I mentioned malaria was a big problem in India. That's where Ross worked. The British Army issued quinine to its soldiers as an anti-malarial drug. The problem is quinine's extremely bitter, and soldiers didn't want to take it, but it was rapidly found that soldiers would drink damn near anything if you just added alcohol. So the quinine was dissolved in water. Water with quinine in it is rather bitter. It's called tonic water. And then if you add alcohol to it, you get the gin and tonic. The origin of the gin and tonic cocktail was as a way to get British soldiers to take their anti-malarial medicine. Be it said, tonic water still does contain quinine, but the stuff you could buy in the supermarket contains too little quinine to be of much good against malaria. The dose is too low to be effective. These days, it's only there for the taste. So yeah, now you know. Not that, I mean, those of you that are under legal drinking age, I'm sure have never tasted such a thing, but when you're old enough, you can try this for yourself. So you can see what's going on there. There you've got a schizont, that red blood cell close to the middle of the slide that has those pink dots in it. Those pink dots are the merozoites that are going to break forth. Now, inside a blood cell, the parasite breaks down up to 80% of the red blood cell's hemoglobin. That leaves behind a compound called heme. I don't know if you've had this or not, but you might remember that hemoglobin is made up of 4 chains of amino acids, right? 2 alpha globins and 2 beta globins. And between the 4 of them, they carry a molecule, it's a rather complex multi-ring molecule called heme. And each heme molecule carries 1 atom of iron. So if the trophozoites are eating the proteins, that leaves free heme behind, and the trophozoites deal with heme by crystallizing it into an inert form called hemozoin. And hemozoin makes these brown crystals. And in this picture, I think you can see the red blood cells themselves are stained very light pink. The merozoites are staining more intense pink, more of a hot pink. And if you look at that infected schizont at about your 8 o'clock position, you can see a thing that is darker brown and not pink. That's that crystal of hemozoin. That's the breakdown product of the free heme that's left behind when the trophozoites eat all your hemoglobin. The reason that matters is that chloroquine blocks the formation of hemozoin. So the heme doesn't form that, and heme is actually toxic to the trophozoites. So with quinine, chloroquine, and drugs like that, the parasites essentially poison themselves because they can't deal with the free heme that their metabolism is leaving behind. They're also looking at, remember that to develop drugs, you want to develop something that hits the pathogen but not the host. Plasmodium has a whole family of enzymes that are not found in animal cells. They're called calcium-dependent protein kinases, CDPKs. In this diagram of the life cycle, you can see those rectangles that say, Merozoite egress CDPK1. Well, calcium-dependent protein kinase 1 is necessary for the merozoites to get out of the schizont. It's also necessary for the schizont, the merozoites, to penetrate red blood cells in the first place. There's another one that is necessary for the male gametes to form, that's CDPK4. CDPK3 is important for the migration of ookinetes, and so on. So these enzymes are necessary for the malaria parasite to transition between stages of the life cycle. They're not found in animal cells. And drugs that are being worked on now are targeting the activity of CDPK, because those enzymes are something that the parasite has that human hosts don't. So if you could figure out a way to block those and only those, you could shut down the parasite life cycle without doing any damage to the host, or at least that's the hope. One of the problems with quinine and chloroquine now is drug-resistant malaria. It's that good old-fashioned natural selection that gets you every time, right? You take the drug and it wipes out 99.9% of the malaria parasites in your body. But if one of them survives and gets mosquito, you can go on to filters and it's too so this is becoming a problem, and currently I believe the frontline drug for malaria treatment was actually developed in China by one of the few Nobel laureates in science who never got a university degree. She grew up during Mao Zedong's Cultural Revolution when the universities were closed, and she basically learned everything by other means. Working with a drug that was long used in Chinese traditional medicine, Artemisia annua, the sweet wormwood, she was able to isolate an anti-malarial compound called artemisinin, which is currently the frontline drug of choice, and I believe we still don't fully understand exactly what artemisinin does. But that has easily saved millions of lives. And then the cheapest way to block the transmission of malaria is still to keep the mosquitoes from biting you in the first place. And beginning in about the year 2000, groups have started handing out insecticide-treated nets, and most importantly, showing people how to use them. I mean, you can't just wrap yourself in one of these nets, because if the net is touching you, the mosquitoes will just bite right through it. So you have to hang them carefully and tuck them the right way to make sure mosquitoes can't get in underneath. But if they're used properly, they'll keep you from getting bitten. And they're credited with bringing malaria deaths down in Africa from an estimated 800,000 to 438,000. The death toll's been cut nearly in half. Sometimes the simplest ways are the best ways, certainly the most cost-effective ways. Insecticide resistance is increasing in mosquito populations, but the nets will still work without insecticide if they're used properly. And the nets can be made locally in Africa or other places where they're needed. You don't need, expensive fabrication facilities to make them. They're cheap. They're $2 to $5 each. And, right now we've certainly got problems of our own that need our donations and, things like that. Certainly if you have some extra money and are thinking about charities to donate to, we've got no shortage of problems within our own borders. But if you feel like it, and if you have the opportunity, there are several charities that provide nets to poor families in Africa. Nothingbutnets.net, I think, has the coolest URL. The CDC Foundation is a private charity that backs up the work of the CDC, and one of the things that they do with the money they gather is provide nets. Againstmalaria.com is another. That might be something worthy of checking out if you have the opportunity. Last cool thing about malaria is I mentioned natural selection. With malaria killing so many people, you would think that natural selection would apply to humans, that many people would die of malaria, those that die would be more likely to pass on their genes, and time we ought to see the rise of malaria resistance in populations where malaria is endemic. Lo and behold, that's just what we do see. There are a number of human genetic variations that confer protection against malaria. And as you'd expect, they are most common in human populations where malaria is endemic. One of them is sickle cell disease. I don't know if you've heard this story, but it does in fact happen to be true. Sickle cell is basically caused by one gene with two alleles. If you are homozygous for the recessive sickle allele, you have sickle cell anemia, which is what's shown here. You see some of the red blood cells are forming not those nice round shapes, but shapes that resemble a sickle, a blade for cutting grain. And episodes of sickling, of lots of blood cells transforming into this shape, can be triggered, and they can cause pretty severe problems. But if you're heterozygous, you have one normal allele and one sickle allele, you don't make very many sickle-celled red blood cells, and you have protection against malaria. And it's been worked out that you would think that sickle cell would be selected against, because if you have this without medical treatment, you're likelier to die young. You're not likely to pass your genes on. But it turns out selection that favors the heterozygotes balances selection against the homozygotes. So the allele stays in the population because people that are heterozygotes have more kids than either homozygous dominant, which have normal blood but risk of getting malaria, and they have more kids than homozygous recessives who have malaria protection but also have sickle cell disease. This, by the way, is called a balanced polymorphism, when selection actually pulls in two different directions at once and ends up coming to a balance. If you take my evolution class one of these days, we will talk about that. We often do. Another blood disease that's similar, that's more common in Asia, where malaria is endemic there, is called thalassemia. Thalassemia is like sickle cell in that if you're homozygous recessive for thalassemia, it's generally fatal eventually, and you tend not to have many kids. But if you have one thalassemia allele and one normal allele, you have protection against malaria. A third one is called glucose-6-phosphate dehydrogenase deficiency. That often has no symptoms at all, but there are certain things that will trigger it. If you have G6PD deficiency, you can lead a normal life, but certain drugs will cause your blood cells to start bursting, and you get an acute attack of anemia with jaundice. Certain drugs will do that. Stress can trigger an attack. And for some reason, fava beans, a species of bean native to Europe, the stuff that Hannibal Lecter ate that guy's liver with, fava beans will actually trigger an attack of anemia and jaundice if you happen to have G6PD deficiency. It's a pretty manageable disease. You just don't eat fava beans. But the point is that having G6-phosphate dehydrogenase deficiency is protective against malaria. There's also one species of Plasmodium, P. vivax. When it binds to red blood cells, it recognizes a surface antigen that happens to be called Duffy. I assume that was the name of the guy that found it, the Duffy glycoprotein. There are two codominant alleles, Duffy A and Duffy B, but individuals that have a mutation in the promoter sequence produce no Duffy at all. They are said to be Duffy null. And the Duffy null genotype is almost unheard of in people of European descent, but it's present in 68% of Africans. And as you can see in the tropical areas of Africa, not the Sahara Desert in northern Africa or the Kalahari in the south, but in the tropics where malaria is most prevalent, somewhere between 90 and 100% of Africans carry this allele. You can see it's also present in parts of South America where malaria is also prevalent. So there are a number of human genes, some of which are disease-causing if you're homozygous for them. And you might wonder what they're doing in the human genome, and it turns out that they have been selected for in populations that are prone to malaria. So malaria has in fact shaped human evolution on the genetic level, which as an evolutionary biologist, I happen to think is pretty neat. Okay, that's what I've got for slides. So I'm going to go ahead and stop recording if you'll give me just a moment. Where's the thing I have to push? There we go. And I will stop recording.